In this video, we're going to be talking about G proteins. And G proteins are ways in which the outside world can communicate with their cells and start a cascade of communication within the cell. It all starts with a ligand binding to a G protein, causes a conformational change, which then allows the GDP, guanosine diphosphate, to be released and guanosine triphosphate to bind to the alpha subunit of the inactive G protein. This activates the G protein, causing the alpha subunit to become dissociated from the beta and the gamma subunits. The activated alpha subunit now binds to an effector protein, and this effector protein will activate other second messengers that will elicit a cellular response. So the G protein provides a link between the receptor that receives the signal and the effector protein that produces the cellular response.
The membrane-bound receptor has a receptor site for a small molecule, called a ligand, exposed on the outside of the cell. The portion of the receptor on the inside of the cell can bind to the G protein, which has a guanosine diphosphate attached to its alpha subunit. When the ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. This step can be repeated as long as the ligand remains bound to the receptor. When the ligand separates from the receptor site, additional G proteins are no longer activated. Inactivation of the alpha subunit occurs when its own phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the subunit. The G protein subunits then recombine and attach to the receptor in the cell membrane.
So there are two effector proteins that activated G protein will bind to. The first of these is adenocyclase. An adenocyclase will activate ATP, the cyclic AMP as you'll hear him pronounce it. And the cyclic AMP activates the protein kinase A, and then protein kinase A phosphates specific proteins. These specific proteins, once they're phosphorylated, will then elicit cellular responses. So adenocyclase is our first effector protein that we look at. Our next is phospholipase C. And so phospholipase C, again, is activated by the alpha subunit of the G protein. Phospholipase C, remember anything with the ASE indicates that it's an enzyme. The phospholipase C then cleaves PIP2, and the PIP2, inositol phosphates, is cleaved into inositol phosphate 3 and DAG. The IP3 can now come down and bound the calcium channels on the endoplasmic reticulum of the cell, opening the calcium channel, allowing calcium to flow out into the cytoplasm. The calcium then binds to a protein and activates it, and then that protein activates the cellular response. A common protein that is bound by calcium is troponin, which is a protein in the skeletal muscle. Activation of troponin allows the muscle to become active, the sliding filament model will work, and then the muscle can contract. So calcium binding to that muscle protein allows the muscle to contract. That is our cellular response because the muscle cell contracts. So you had a question in your homework. Well, we'll get to that in just a minute. Let's watch this video first. There's your question, and let's watch the video.
When a signal molecule, such as epinephrine, binds to a cell surface receptor protein, it activates a G protein on the inside of the cell. The G protein then stimulates adenylyl cyclase to produce large amounts of cyclic AMP within the cell. The cyclic AMP then binds to and activates a target protein such as alpha kinase, which adds phosphates to specific proteins in the cell. The effect of this phosphorylation depends on the identity of the cell and the proteins that are phosphorylated. Calcium ions also serve as second messengers. In response to a signal molecule from other cells, the cell surface receptor protein activates a G protein, which in turn activates the enzyme phospholipase C. This enzyme catalyzes the production of inositol triphosphate, which diffuses through the cytoplasm to the endoplasmic reticulum. The inositol triphosphate binds to and opens the calcium channels, allowing calcium ions to flow from the endoplasmic reticulum into the cytoplasm. The calcium ions trigger proteins sensitive to calcium to initiate a variety of activities.
Okay, so one of your questions in the homework too that you have is how the effect of a phospholipase C inhibitor, and remember phospholipase C is one of our effector molecules that the G proteins bind to, on the fight or flight response. And so you eat something in the jungle because you are feeling hungry. You decide to snack on some of the local plants as they are delicious. Unbeknownst to you, the plant you decide to eat contains a compound that acts as an inhibitor of phospholipase C activity. Remember, phospholipase C activity cleaves PIP2 to inositol triphosphate and into DAG. Soon after consuming your snack, you begin to hear a rustle in the leaves. You fear that it could be a large carnivorous animal and thus choose to run. And as I had mentioned before, that calcium that is released in response to the inositol triphosphate binding to the calcium channel, that calcium is responsible for your skeletal muscle contractions. So if you want to run, you need that calcium so that you can contract the muscles. The epinephrine release stimulated by your fight or flight response leads to the activation of a specific G protein coupled receptor. So that is our GPCR, G protein coupled receptor, that stimulate the breakdown of macromolecules to generate the energy you need to run. And so that energy again is in the form of muscle contraction. What effect will the presence of a phospholipase C inhibitor have on your ability to escape the predator? Select all that apply. So, will it prevent the formation of cyclic AMP? No, because cyclic AMP is activated by adenylate cyclase, which is a different effector protein that the G protein binds with. So this would be no. Will it prevent the formation of IP3, inositol triphosphate? Yes, it will, because you're inhibiting phospholipase C, which cleaves the PIP2 into IP3 and DAG, so it'll also prevent the formation of DAG. So it'll prevent the formation of these two, but will have no effect on cyclic AMP. Cyclic AMP, remember, now activates protein kinase A, so it'll have no effect on the activation of protein kinase A because you didn't affect cyclic AMP. It will likely inhibit some of the signaling pathways downstream of our G protein coupled receptors activated by epinephrine. Yes, because we saw that two effector proteins, one is cyclic AMP, which is formed by adenyl cyclase, and two was our phospholipase C. So it'll affect the phospholipase C pathways, but not the adenylate cyclase pathways. So yes, it will inhibit some of our signaling pathways, but not all of them because adenylate cyclase is still functional. It will prevent the activation of the heterotrimeric G protein involved in the signaling pathway. No, because this is beforehand. So this is where the alpha and beta and gamma bind with GTP and then become activated. So this is before phospholipase C, so no, it will not be affected. So again, phospholipase C cleaves PIP2 to DAG and IP3, so these two will be affected. IP3 can then diffuse through the cytoplasm to activate the channel length receptors, which leads to release of calcium. But that is not going to happen because you have the phospholipase C inhibitor. And since phospholipase C is downstream of the GPCRs, your G protein coupled receptors, inhibiting this enzyme will not inhibit all of the GPCRs because you're only inhibiting those that involve phospholipase C. The heterotrimeric G protein should still be activated by the receptor binding to the epinephrine, and cyclic AMP production and PKT activity should not be affected by a phospholipase C because they are dependent on adenylate cyclase and not phospholipase C inhibitor. Hope this helps, and if you have any questions, please let me know.